so this method can facilitate the establishment of biomarkers for skin diseases and also reduce the risk for clinical research participants. The main advantages of this technique are that it does not require intensive training and it does not leave any scars.
"5 minutes before the microbiopsy, place an empty 2 ml microcentrifuge tube on dry ice for 5 minutes. Wearing disposable gloves, spray the hands and tools with 70% ethanol. Before sanitizing the application site on the subject with an alcohol wipe. Remove the microbiopsy from the sterilized gamma radiation package without touching the microneedle, and pull the plunger until a click is heard and the plunger locks in place. Aim the loaded device at an approximately perpendicular angle to the skin to be sampled, taking care that the sampling area is in a fixed position, and apply the device onto the skin within at least 1 kilogram of force. Then press the trigger and hold the device in place for at least 10 seconds.
If the device is released before 10 seconds, there's a higher chance of variation in the blood adsorption and also the subsequent analysis.
When the sample has been absorbed, gently pull the plunger and absorbent microneedle from the case and use sterile forceps to pull on the 2 holes of the microneedle to remove it from the plunger. Add 50 microliters of extraction buffer to the tube on dry ice and place the intact microneedle in the tube on dry ice, needle side down.
Be careful not to let the tip of the microneedle touch anything during the removal process.
Vortex the tube for 3